Collect the water sample to be analysed. Ensure to follow proper sampling technique: disinfection, flushing, and using the sterile bottle. Take the luminometer, remove the tube holder, and power on the device. Calibration will start automatically. Before beginning the analysis, ensure all reagents have reached room temperature. Take a syringe and remove the plunger. Open the filter blister pack and screw the filter onto the syringe. Place the plastic blister cup in the holder. Pour the water sample into the syringe. Note the sample volume. Filter the entire sample. Once there is no more water passing through, stop applying pressure. Add 4 drops of extractant into the plastic blister cup. Insert the syringe tip into the plastic blister cup and draw up all the reagent. Open the LumiTube foil pouch and remove a tube. Carefully reseal the foil pouch after use. Remove the cap using the tube holder and place the LumiTube on the blue rack. Place the filter tip into the LumiTube. Push the plunger with the palm of your hand until a white foam appears. Then stop pressing to avoid filling the tube with foam. Attach the LumiTube to the tube holder. Gently shake it in a pendulum motion, then insert it into the luminometer. Press the Start button to take the measurement. After 10 seconds, the R1 result will be displayed. Remove the LumiTube. Add a drop of standard, holding the dropper 1 centimeter above the tube. Mix using a pendulum motion and by tapping the tube on a flat surface. Insert the assembly into the luminometer and press the Start button to record the measurement. After 10 seconds, the R2 result will be displayed. To interpret the data, use the Lumen application. Enter the filtered volume and the R1 and R2 results. The application automatically calculates the result, records the data, and provides a clear interpretation, with the option to edit a report. You can also supplement this information with your other operational parameters: biocides, pH, turbidity, conductivity, etc.